Hi everyone, my name is Sonia. In this video, we are going to discuss about electron microscopy. An electron microscope uses a beam of electrons to illuminate a specimen and produce a magnified image. It is used for those objects which are smaller than 0.2 micrometer. An electron microscope has greater resolving power than a light-powered optical microscope because electrons have wavelengths about 1 lakh times shorter than visible light photons. It produces black and white images. History of electron microscope. The first electron microscope was developed in 1931 by Ernest Ruska and Max Knoll. Basic principle of electron microscope. Electron microscopes use signals arising from the interaction of an electron beam with the sample to obtain information about structure, morphology and composition. Beam of electrons passes through the specimen, then through a series of lenses that magnify the image. The working principle of electron microscope. The electron gun generates electrons. Two sets of condenser lenses focus the electron beam on the specimen and then into thin beam. To move electrons down the column, an accelerating voltage is applied between tungsten filament and anode. The specimen to be examined is made extremely thin, at least 200 times thinner than those used in the optical microscope. Ultra-thin sections of 20 to 100 nanometer are cut, which is already placed on the specimen holder. The electronic beam passes through the specimen and electrons are scattered depending upon the thickness or refractive index of different parts of the specimen. The denser regions in the specimen scatter more electrons and therefore appear darker in the image, since fewer electrons strike that area of the screen. In contrast, transparent regions are brighter. The electron beam coming out of the specimen passes to the objective lens, which has high power and forms the intermediate magnified image. Ocular lenses then produce the final further magnified image. There are two types of electron microscopes used in clinical and biomedical research settings: the transmission electron microscope and scanning electron microscope. Differences between scanning electron microscope and transmission electron microscope. Scanning electron microscope is based on scattered electrons. Transmission electron microscope is based on transmitted electrons. In scanning electron microscope, large amount of sample to be analyzed. Transmission electron microscope, only small amount of sample can be analyzed. Scanning electron microscope used to study the topography of materials. It provides detailed images of the surfaces of cells and whole organisms. Transmission electron microscope is used to thin specimens through which electrons can pass, generating a projection image. In scanning electron microscope, resolution is small, much higher resolution in transmission electron microscope. Scanning electron microscope produces 3D images. Transmission electron microscope produces 2D image. Parts of electron microscope. Electron microscope is in the form of a tall vacuum column that is vertically mounted. It has the following components. Electron gun. The electron gun is a heated tungsten filament which generates electrons. Another component is electromagnetic lenses. The condenser lens focuses the electron beam on the specimen. A second condenser lens forms the electrons into a thin, tight beam. The electron beam coming out of the specimen passes down the second of magnetic coils called the objective lens, which has high power and forms the intermediate magnified image. The third set of magnetic lenses called projector or ocular lenses produce the final further magnified image. Another component is specimen holder. The specimen holder is an extremely thin film of carbon held by a metal grid. Image viewing and recording system. The final image is projected on a fluorescent screen. Below the fluorescent screen is a camera for recording the image. This is an image of parts of electron microscope. Differences between light microscope and electron microscope. In light microscope, light is the illuminating source. In electron microscope, the beam of electrons is the electron source. In light microscope, specimen preparation takes usually few minutes to hours. Live or dead specimen may be seen. In electron microscope, specimen preparation takes usually a few days. Only dead or dried specimens are seen. In light microscope, condenser, objective and eyepiece lenses are made up of glasses. In electron microscope, all lenses are electromagnetic. In light microscope, specimen is stained by colored dyes. In electron microscope, specimen is coated with heavy metals in order to reflect electrons. In light microscope has low resolving power. It has a magnification of 500x to 1500x. In light microscope, vacuum is not required. In electron microscope, vacuum is essential for its operation. In light microscope, image is seen by eyes through ocular lens. In electron microscope, image is produced on fluorescent screen or photographic plate. Here we can see an image of light microscope and electron microscope and its differences. Applications of electron microscope. Electron microscopes are used to investigate the ultrastructure of a wide range of biological and inorganic specimens, including microorganisms, cells, large molecules, biopsy samples, metals, and crystals. Industrially, electron microscopes are often used for quality control and failure analysis. Science of microbiology use electron microscope to study of microorganisms like bacteria, virus and other pathogens have made the treatment of diseases very effective. Preparation of specimens for electron microscopy. There are many steps in specimen preparation. Cleaning the surface of the specimen, stabilizing, rinsing, dehydrating, sectioning, freeze etching or freeze fracture, mounting or embedding, metal shadowing or coating. First step, cleaning the surface of the specimen. Surface contains many unwanted deposits such as dust, mud, soil. Next step is stabilizing the specimen. Hard dry materials such as wood, feathers, dried insects or shells can be examined with little further treatment. But living cells and tissues and all soft-bodied organisms usually require chemical fixation to preserve and stabilize their structure. Fixation is the another step. Fixation is done by incubating the specimen in a solution of a buffered chemical fixative. Formaldehyde and tannic acid are commonly used fixatives. Another step is rinsing the specimen. Sample must be rinsed to remove excessive fixatives, usually rinsed using sterile water. Next step is dehydrating the specimens. Water must be removed. Dehydration is performed with ethanol or acetone. Another step is sectioning. Sectioning is done by ultramicrotome. Specimen is sliced using a glass or diamond knife. Slices will be semi-transparent to electrons. Section will have 60 to 90 nanometer thickness only. Freeze etching is the sublimation of surface ice under vacuum to reveal details of the fractured face that were originally hidden. It is used to investigate for indents, cell organelles, membranes, layers and emulsions. Another step is mounting or embedding. Dehydrated specimen has to be embedded or mounted on the holder. Embedding is done by using an adhesive such as epoxy resin or acrylic resin. Embedding is mostly done for transmission electron microscope, not scanning electron microscope. Another step is metal shadowing or coating the specimen. Metal shadowing is used to increase the conductivity of the specimen. Coated with thin layer, that is 20 nanometer to 30 nanometer of conductive metal. Conductive materials are gold, platinum, iridium, graphite, chromium, tungsten.
Fixatives. So the fixation to kill a biological specimen but protect its native structure. So there is chemical fixation and cryofixation. So first we discussing about the chemical fixation. Coagulant fixatives, cross-linking fixatives, compound fixatives are the chemical fixators. Ethanol, methanol, acetone, acetic acid, picric acid, tetrachloroacetic acid, these are the coagulant fixators. Formaldehyde, glutaraldehyde, mercury chloride, osmium tetroxide, chloral hydrate, these are the cross-linking fixators. Alcohol formalin and formaldehyde, glutaraldehyde are compound fixators. So formaldehyde and glutaraldehyde are mostly commonly used fixatives. So first we are discussing about the formaldehyde. It is stored as an aqueous solution. This aqueous solution form methyl hydrate. This reacts side chains of protein form reactive hydromethyl side chain. Formaldehyde react with nuclear protein and nucleic acid that stabilize nucleic acid protein shell and modify nucleotide by reacting with the free amino acid group as it does with protein. The side chains of peptide or protein react with methylene, the methylene hydrate, hence high affinity for formaldehyde. Then next is the glutaraldehyde. It is 5-carbon dialdehyde, most widely applied in fixatives in SEM and TEM. Glutaraldehyde and acrolein cause extensive, rapid and permanent cross-linking of the proteins. So there are 2 aldehyde group on the both end of the molecule. So this unreacted aldehyde group is cross-linked with the protein. So this extensive cross-linking preserve the ultra structure. Secondary fixators. To increase the contrast of minute structure, we use the secondary fixation. It is done by osmium tetroxide. So the osmium tetroxide stabilize many protein by transforming it to gel without destroying its structure. It is most used in TEM. The second one is the cryo preservation. In cryo preservation, very low temperature is used to preserve living cells and tissue and maintain their viability. It is mostly used in TEM. Vitrification. Water becomes frozen liquid state, so ice is not formed. This state can be maintained for a long time. Thank you.